Alright, so next we're going to be looking at the sliding filament model of contraction. Here's a brief introduction into this model, and then we're going to take a step back and talk about the initial stimulus that leads to the sliding filament model, which includes the cross-bridge cycle. This model states that during contraction, the thin and thick filaments slide past each other. The greater overlap between the actin and myosin is going to allow sarcomeres to shorten, which then allows for myofibrils to shorten, and the shortening of myofibrils mean that cells are going to shorten. And if all the cells in a muscle as an organ shorten, then the muscle organ itself is also going to shorten. And shortening of a muscle is what we see in muscle contraction. This requires cross-bridge attachments to be formed, and cross-bridge attachments can only form if the troponin and tropomyosin complex that covers up the actin active site has been moved out of the way. This is possible if calcium intracellular concentrations have increased, because calcium is the final go signal for contraction, and when it binds to the troponin of the tropomyosin troponin complex, then we see that the actin active sites are revealed, and myosin of the thick filament has its heads bind to the actin active site. What we see as a muscle cell shortens are the I-band shorten, the distance between the Z-discs shorten, the H-zones disappear, and the contiguous A-bands move closer together. Keep in mind that the myofilaments do not have their lengths shorten. The length of the myofilaments, actin and myosin, do not change. We will just see greater overlap. Alright, so that's kind of a brief introduction for what we're working towards, which is the cross-bridge cycle where myosin and actin bind. But let's take a step back and start at the beginning with the initial stimulus for muscle contraction. So we're going to be looking more now at the physiology of skeletal muscle fibers. It all begins with a nerve stimulus. This is a nerve stimulus coming from a somatic nervous system motor neuron. This output signal travels to skeletal muscle, and the location where the axonal branch or axon terminals of the somatic nervous system motor neuron almost touch a single muscle fiber is called the neuromuscular junction. Axons are the long process or long projection off of the soma or cell body of a neuron, and these long processes are able to carry electrical signals called action potentials. It's these action potentials that must travel to the sarcolemma of a single skeletal muscle fiber in order for calcium to eventually be released and therefore a cross bridge to be formed. With the sliding filament model of contraction, we want to define contraction in order to talk about it a little more succinctly. Physiologists specifically define contraction really just in terms of the activation of the myosin cross bridges. So even though we initiate this process with a motor neuron sending its electrical activity to a skeletal muscle fiber, it's only the end product, the force-generating sites, that we actually use to define what's happening when we say a muscle contracts. That's why the relationship between the thick and thin filaments is very important, especially in describing how contraction proceeds and how we get not only sarcomere shortening, but eventually cell shortening and muscle as an organ shortening. So the sliding filament model of contraction could also be thought of as the cross-bridge cycle. A cross-bridge is when myosin binds to actin, and that causes increased overlap between the two myofilaments. Again, this is a cyclic process, and it all begins during excitation-contraction coupling when calcium is released from the sarcoplasmic reticulum. It's because the calcium released from the sarcoplasmic reticulum can bind to the troponin of the tropomyosin troponin cascade that allows the actin active site to bind to a myosin head and cause the shortening that we associate with the sliding filament model of contraction. ATP is also utilized during this process, so it's an active process. So here's some general information about contraction as we move forward and look at the physiology. We want to generate tension, but just because we generate tension doesn't mean we cause shortening. For example, if you were to walk up to a wall in your house and try to push over the wall, you couldn't do it, or hopefully you don't do it. And that is because you're generating tension in those muscles, but you're not generating enough force to cause contraction, therefore you can't push over the wall. So just because we get tension doesn't mean we get contraction, an actual movement of what we're trying to move. There are plenty of steps that generate tension, and then the final step that generates contraction would be the cross-bridge cycle. The series of steps that begins with that excitation of the motor neuron jumping to the sarcolemma is called the neuromuscular junction. It's followed by calcium release from the sarcoplasmic reticulum, which is a part of excitation-contraction coupling, And then, of course, we end with the cross-bridge cycle, a contraction cycle, which is going to be a thin-thick filament reaction. Muscle fiber contractions can be described of two types, isotonic and isometric. So we'll look at that as well, and we'll make sure to point out the differences again between tension and actual shortening of the sarcomere. Again, just because we have tension doesn't mean that we cause shortening of the muscle fiber, which is the muscle cell. And we have to have that tension generated by cross bridges repeatedly, so myosin on actin, or the thick on thin filaments, and we have to have that tension basically exceed the force opposing shortening in order for actual movement to occur. So what's inducing skeletal muscle contraction? Let's start all the way back at our nervous system stimulation again. That's because our first step is at the neuromuscular junction. So this is activation at the neuromuscular junction, where the fiber must be activated via a change in membrane potential. This is a charge, so we have to have that charge jump from the motor neuron to the sarcolemma of the skeletal muscle fiber. The sarcolemma must generate an electrical current, which is an action potential, in order for that action potential or electrical current to travel into the sarcoplasm via the T-tubules. So at the neuromuscular junction, it is paramount that the action potential of the axon of the motor neuron jumps to the sarcolemma, specifically at the location where the neuron meets the muscle. That's when we're calling that the neuromuscular junction. The action potential then is going to travel throughout the sarcolemma to a T-tubule. Via the T-tubules, which are connected to the sarcolemma, that electrical action potential is able to travel into the cell, and that will cause the sarcoplasmic reticulum to release calcium into the sarcoplasm. This will cause a rise in intracellular calcium levels. And remember, calcium is the final go signal for contraction. So the release of calcium is essentially a way that we can link that electrical signal that had to travel through the T-tubule to contraction. So this is phase 2, excitation-contraction coupling, where we're again linking the electrical signal to contraction by using the electrical signal to cause the sarcoplasmic reticulum to release calcium, and because calcium is the final go signal for contraction, that's enough to link the two. So again, step 1, action potential has to travel from neuron to muscle. Step 2, action potential has to travel along the muscle cell sarcolemma into the cell, causing the release of calcium from the sarcoplasmic reticulum. And calcium as the final go signal for contraction is enough to cause the cross-bridge cycle to begin. Alright, so let's talk about phase 1 and 2, neuromuscular junction, and excitation-contraction coupling in more detail. So again, we went all the way back to step 1. The neuromuscular junction is talking about getting that action potential from the motor neuron to the muscle cell. And then step 2, excitation-contraction coupling, is using the action potential once it's in the muscle cell to cause the release of calcium. And since calcium is the final go signal for contraction because it initiates the cross-bridge cycle, that's enough to couple or connect the excitation signal, the action potential, with a contraction phase, which is going to be the cross-bridge cycle. Okay. During phase 1, we know we're getting the action potential to go from the motor neuron to the muscle fiber. To do this, the action potential, the electrical signal, has to arrive at the end of the axon. The axon is the long process off a neuron. And when it gets to the end of the axon, it goes into axon terminals, which are branches found at the end of an axon. The axon terminals are located at the neuromuscular junction, which is the location close to the skeletal muscle fiber. Also notice I keep saying close to because the axon terminals do not touch the sarcolemma. The axon terminals contain vesicles filled with acetylcholine. They release their acetylcholine due to an influx of calcium at the axon terminals. The acetylcholine travels into the space between the axon terminals and the sarcolemma. The acetylcholine then travels across the space and binds to receptors that recognize acetylcholine. Acetylcholine bound to receptors on the sarcolemma causes channels to open in the membrane of the sarcolemma. This increases ion permeability of the sarcolemma and allows for an influx of ions, which causes depolarization or a membrane potential change. This local membrane potential change, or local depolarization, called an in-plate potential, is what initiates the action potential in the sarcolemma. Once the electrical action potential is in the sarcolemma, it can travel across the entire sarcolemma, so now we're in phase 2. Once the sarcolemma's electrical signal reaches the T-tubules, it travels into the sarcoplasm via these T-tubules. The T-tubules touch the sarcoplasmic reticulum at a triad, so it touches 2 terminal cisternae. The sarcoplasmic reticulum contains calcium, and when the action potential travels along the T-tubules, which touch the sarcoplasmic reticulum, it causes the sarcoplasmic reticulum to release calcium, which causes a rise in intracellular calcium levels. And a rise in intracellular calcium levels means that there is calcium in the sarcoplasm, and that means that it can bind to troponin. Troponin binding to calcium causes it to twist and move, moving the troponin-tropomyosin cascade out of the way and revealing the actin active sites and allowing myosin to bind to actin. When myosin binds to actin, that initiates phase 3, which is the cross-bridge cycle. So let's look at the anatomy of the neuromuscular junction. So we have a motor neuron. It has a long projection off of the cell body of this neuron called an axon. At the end of the axon, there are branches, and these branches are called axon terminals. And each individual branch at the end has an axon terminal. So that's why initially I said it as plural. So all of these branches are axon terminals. One singular branch has a bud at the end called an axon terminal. There is a space between the axon terminal itself and the sarcolemma, and that space is called the synaptic cleft. The area where the axon terminal gets really close to the sarcolemma contains folds called junctional folds. The junctional folds of the sarcolemma are many, and they are located along a location called an end plate. So that we have the axon ending that gives off several curling branches, and that is collectively forming the neuromuscular junction where it gets close to the sarcolemma at locations called junctional folds. But the axon terminal never touches the junctional folds, and that space between them is the synaptic cleft. So if you're asked to list the 3 parts of a neuromuscular junction, those would be the axon terminal, synaptic cleft, and the junctional folds. You may also put the junctional folds at the end plate or just end plate instead of junctional folds. But these are the 3 parts of the neuromuscular junction. The axonal endings that we're calling the terminal branches or axon terminals of the motor neuron, they contain synaptic vesicles. Let's go back. These little bubbles here, they are synaptic vesicles, and in them is acetylcholine, which is a neurotransmitter. Alright, so the axonal endings of each neuron contain synaptic vesicles, which contain the neurotransmitter acetylcholine. Acetylcholine is a neurotransmitter that wants to bind to receptors on the junctional folds of the sarcolemma. So acetylcholine actually has to be secreted from the synaptic vesicles via exocytosis and travel across the synaptic cleft, or that space, in order to get to the receptors on the junctional folds of the sarcolemma. So again, the receptors for acetylcholine are on the sarcolemma. The vesicles of acetylcholine are in the axon terminals, so there's exocytosis and travel in order to get the acetylcholine to bind to the receptors for themselves on the sarcolemma. So let's look at those steps. So again, here's just another view of the neuromuscular junction. Even though it's showing you how close the axon terminals are to the sarcolemma, keep in mind they do not touch. There is a space. That space is the synaptic cleft. So axon terminals, synaptic cleft, and junctional folds of the sarcolemma, that's what makes up a neuromuscular junction. So the steps here are the electrical signal that we're calling an action potential travels down the axon to its end. It travels to each of the individual axon terminals. Within the axon terminals are synaptic vesicles that contain acetylcholine. When the electrical signal, the action potential, reaches an axon terminal, it opens voltage-gated calcium channels. When calcium goes in to the axon terminal, acetylcholine goes out. That's the trigger. Calcium goes in to the axon terminal, acetylcholine goes out. And the ion channel that allows calcium in, it's voltage gated. So when the volt or the electricity reaches the axon terminal, that's when these channels open. When acetylcholine leaves via exocytosis, it travels across the small synaptic cleft. The synaptic cleft, again, is the space. And as the acetylcholine leaves via exocytosis and travels across the cleft, it encounters receptors here in kind of dark purple, and the receptors bind acetylcholine. When these receptors bind acetylcholine, they open because the receptors are essentially closed channels, and acetylcholine is the trigger to open these channels that are found in the junctional folds of the sarcolemma. When these channels open, ions can move in and out. More ions with a charge move in than move out, and this changes the membrane potential or volt across the membrane of the skeletal muscle cell. This change in voltage across the membrane creates a membrane potential called a local depolarization or end plate potential. And we'll talk more about this later, but acetylcholine is recycled and broken down via an enzyme that's also in the synaptic cleft called the acetylcholine esterase enzyme. Alright, so let's go through that again. So how does a motor neuron stimulate a skeletal muscle fiber at the neuromuscular junction? First, nerve impulse reaches the axon terminals. Second, impulse reaching the axon terminals opens voltage-gated calcium channels. Once those calcium channels open in the axon terminal, calcium goes in, acetylcholine goes out. Calcium in, acetylcholine out. Acetylcholine would leave the axon terminals via exocytosis. Once acetylcholine enters the synaptic cleft, it diffuses across the cleft, binds with its receptors, called acetylcholine receptors, on the junctional folds of the sarcolemma. This is the initiation of the action potential jumping to the sarcolemma, because when acetylcholine binds to its receptors on those junctional folds, then it opens ion channels that allow sodium ions to come into the cell, creating a local depolarization as potassium ions leave. This local depolarization is a change in membrane potential called the end plate potential. And at this point, I would watch the A&P Flicks video on muscle contraction. The name of the video is Muscle Contraction Part 1, Events at the Neuromuscular Junction, and you can find a link to this video in your Blackboard announcements. I would pause this video, watch the A&P Flicks video, and then come back. Alright, let's just review one more time, now that you've watched the video, of the excitation at the neuromuscular junction. So this will be a much quicker review since you just watched that video. Alright, step 1, action potential reaches the axon terminal, opens voltage-gated calcium channels, calcium in, acetylcholine out, acetylcholine exits via exocytosis, acetylcholine travels across the synaptic cleft to bind to acetylcholine receptors. Acetylcholine receptors are channels that are closed. When acetylcholine binds to these channels, they open, allowing an influx of sodium and an efflux of potassium. And when we have an influx of sodium, it creates depolarization. This is happening in just this location, so it's a local depolarization called an end plate potential. And then last but not least, acetylcholine is removed or broken down. It is terminated by acetylcholine esterase, so it would break down into its components, and some of it can be recycled. We don't want acetylcholine to remain bound to the acetylcholine receptors on the junctional fold of the sarcolemma, because that would cause the sarcolemma or that muscle cell to continue to be stimulated. And we couldn't stop contraction and restart contraction if the stimulation was continuous. It would cause tetanus. And so we don't want to block new impulses from reaching the muscle cells. It would make it really difficult to control muscle cell activity. So you want to make sure we're breaking down acetylcholine by either allowing it to diffuse away or breaking it down with the enzyme acetylcholine esterase. The location of acetylcholine esterase in this breakdown would be in the synaptic cleft. And our next step is going to be the excitation contraction coupling step. Alright, the next step is excitation contraction coupling. So this is after the neuromuscular junction. After we've gotten the action potential into the sarcolemma, it's going to propagate or move along the sarcolemma to the T-tubule. And because the T-tubule is continuous or connected to the sarcolemma, that action potential can just flow from the sarcolemma to the T-tubule, and the T-tubule goes within the cell. So we're able to bring this electrical signal from outside or along the sarcolemma to the inside of the cell. And T-tubules touch or abut the endoplasmic reticulum that's in a muscle cell called the sarcoplasmic reticulum. Specifically, a T-tubule is going to touch the terminal cisternae at the triad location inside a muscle cell. This is important because the sarcoplasmic reticulum is where calcium is stored in a skeletal muscle cell. And when the T-tubule brings its action potential that it got from the sarcolemma, that causes the sarcoplasmic reticulum to release calcium. And the release of calcium is the final go signal for contraction because it initiates the contraction cycle called the cross-bridge cycle, which includes attachment, pivot, detachment, and return of the myosin and actin heads as they move and overlap more, causing a sarcomere to shorten, which eventually leads to cell shortening. At this point, I would go watch the excitation contraction coupling video. And so I would pause this video, go watch the A&P Flicks muscle contraction part 2, excitation contraction coupling, and then come back to this video where we'll review. So you should have noticed from that video you just watched that the action potential is able to move into the cell because of the T-tubule. Notice how they're connected. And that at the triad, where the two terminal cisternae of the sarcoplasmic reticulum are located next to the T-tubule, it is the propagation of the action potential inside the cell that opens an ion channel that allows for the release of calcium from the sarcoplasmic reticulum. So again, it's the sarcoplasmic reticulum holding on to calcium, and we need to open a voltage-gated channel to let it enter into the sarcoplasm. And that voltage arrives via the T-tubule, opens the voltage-gated calcium channel, and calcium is allowed to enter the sarcoplasm, and we see a rise in intracellular calcium levels. The reason this is going to link or couple excitation to contraction is it's the voltage, the action potential, or the excitation traveling from the sarcolemma to the T-tubule that causes the release of calcium from the sarcoplasmic reticulum, which is the final go signal for contraction. So it's a short phase, but this is how we link the voltage or excitation to contraction or calcium release. And calcium is important because it binds to troponin of the troponin-tropomyosin cascade. Troponin rotates and moves the tropomyosin out of the way, revealing the actin active sites for myosin to bind. And when myosin binds to actin, we can have a cross bridge. And the cross bridge cycle is how physiologists describe muscle contraction because it actually causes shortening of sarcomeres, therefore myofibrils, therefore cells, and eventually muscles as an organ. So since we're moving into the cross bridge, let's just review what we know of the sliding filament model of contraction. Actin is called a thin filament. Myosin is also called a thick filament. Actin and myosin are thin, thick filaments. Those are the myofilaments found within myofibrils. Sarcomeres are units of myofibrils, so therefore they also contain actin and myosin. And we're going to see that during the cross-bridge cycle, which uses the sliding filament model of contraction, we'll get greater overlap between actin and myosin, even though the filaments do not change length. They just relapse or cover each other more. So they're going to overlap to a greater degree, but the filaments themselves never shorten. In a relaxed state, the thin and thick filaments overlap only slightly. Therefore, we have dark A bands and light I bands, but they always overlap. So they're never going to completely not overlap, but we will see more overlap during the sliding filament model of contraction when the cross bridge is initiated. The stimulation required for cross bridge formation is the release of calcium from the sarcoplasmic reticulum. The sarcoplasmic reticulum binds to troponin, which rotates, and that moves tropomyosin out of the way, allowing for myosin heads to bind to actin and initiating that cross bridge. Each myosin head is going to bind and detach from the actin active site multiple times, so it really pivots and acts like a ratchet to generate tension and propel the actin myofilament closer to the center of a sarcomere. So we have the myosin heads pivoting and creating tension and moving the actin closer to the center of a sarcomere, and this shortens the sarcomere. So again, we're getting more overlap, not shortening of the myofilaments. So during this event, sarcomeres, which are units of a myofibril, shorten. When all sarcomeres in a myofibril shorten, the whole myofibril shortens. And then this can lead up to the entire muscle organ shortening or contraction of that muscle organ as its cells shorten because of the myofibril and sarcomere shortening. So let's look at the contraction cycle. So what we're looking at first is we have ATP bind to a myosin head. ATP then gets broken down, and that cocks the myosin head, so that's going to bring it back into what we call the cocked position. We broke down ATP into adenosine diphosphate, or ADP, an inorganic phosphate. That release of energy from the dephosphorylation of ATP was enough to put myosin in this cocked position, and then allow it to be ready next to bind to actin active site. So we have ATP binds, is broken down, which puts myosin in the cocked position, which is a high energy form. As that high energy form deteriorates, myosin binds to actin. Then the inorganic phosphate is going to be released. And when inorganic phosphate is released from the myosin head, that causes the power stroke or the pivot, where myosin moves forward, thereby moving actin forward or towards the center of the sarcomere. Next, the adenosine diphosphate is also going to be released. And when the adenosine diphosphate is released, we have the completely lowest energy form of myosin, and it's not going to be able to do much else or detach and then start over again until ATP binds. So we'll get a little bit of detachment right when ATP binds, and then ATP again will be broken down, it'll bind to actin, it'll pivot, it'll be in a low energy form, it'll bind to ATP, detach, cock, build up to a slightly higher energy form, and bind to actin. Let's look at the steps sequentially. Keep in mind that even though we're doing this sequentially, that these steps are a cycle, so they just repeat when we're done. So we'll actually start here with cross-bridge formation, so myosin binds to actin. Once myosin binds to actin, then we have the power stroke, and that happened when inorganic phosphate was removed from the myosin head. The power stroke moves the actin filament toward the M line of a sarcomere. Then, because ATP binds, eventually again, to the myosin head, we get removal of myosin from actin. So this is cross-bridge detachment. And then when ATP is hydrolyzed via the removal of a phosphate, creating ADP and inorganic phosphate, that causes the recocking of the myosin head so that it's in its highest energy state, and then again it will form a cross bridge, power stroke, detach, and then again we will have the recocking of the myosin head. So this just repeats. At this point, I recommend you watch the A&P Flicks Muscle Contraction Part 3 Cross Bridge Cycle video. Again, these videos are attached to a Blackboard announcement. So make sure that you watch this video. They are also available if you would like to Google them on YouTube. Alright, so make sure you watch that video. At this point, I may even recommend that you watch all 3 videos again. I also recommend that you review the steps, which include the neuromuscular junction, excitation, contraction, coupling, and cross-bridge cycle, which are the steps of skeletal muscle contraction. Again, neuromuscular junction, excitation, contraction, coupling, cross-bridge cycle. Those are the steps of skeletal muscle contraction. So please go back and review the information in your book, in those videos, but for proceeding forward. You can also type this link into a web browser and it'll take you to an animation that goes through those steps. So the neuromuscular junction, excitation contraction coupling, and the cross bridge cycle. Make sure to review those and understand them before proceeding forward.